Hi, everybody. Welcome to the Chats with Children. I hope you're safe and well and happy new year. I hope you've had a good start to the year. Today, I'm delighted to be joined by Justin Neill, who is the Chief Technology Officer at BrightSpec. And today, we're going to be learning all about molecular rotational resonance, or MRR. It might be easy to say it the long way around, actually, rather than MRR technology. We're going to learn more about it, how it works, what its applications are in pharma, and what its future is. So Justin's going to tell me all about that. But first of all, Justin, how are you?
Doing great, doing great. Thank you very much for the invitation and great to talk to you. How are you?
Well, I'm very, very well. It's lovely to talk to you and learn more about MRR. But before we do that, for people who are not familiar with BrightSpec, perhaps you give us a very quick overview of
Mhm.
who the company are and what you do as a business.
Sure. Yeah. So BrightSpec was founded in originally in 2013 to commercialize development out of the University of Virginia, so Charlottesville, Virginia, which is where our R&D headquarters is based, to basically develop this technology, this MRR technique from the research, its research background into commercial products, which we'll get into over through this chat. That's really what that was our organizing mission, and we've developed quite a bit of technology and products over the years that are leading us to the current day.
Fantastic. Well, look, let's get into it then. So, obviously, for people not familiar with MRR, tell us more about the technology itself and how it works.
Sure. Yeah. So MRR is so we're characterizing molecules, small molecules in the gas phase through their molecular rotation. So just like it's a form of spectroscopy. So just like other types of spectroscopy, NMR, infrared spectroscopy, we're taking radiation, light, and letting it interact with the molecular sample that we've prepared and looking at the response and the results to understand more about the sample that's been introduced. So in MRR, what we're looking at is the molecule's rotation.
Right.
So, and it's really when you have either a molecule or even an object, its end over end rotation in space is going to really be governed by its moment of inertia, the distribution of its mass
Right.
around its axes. And in particular, if you change that, if you rearrange the atoms into different configurations, look at things like isomers, uh, that will affect the speeds at which that rotation can happen, and we can characterize molecules on that basis. So what we do basically, we're using microwave to radio frequency light interacting with samples in the gas phase, and then viewing, after we excite them, they'll resonate. That second R in MRR, they'll rotate at very specific speeds and emit radiation at highly characteristic points in the spectrum, and we can record that information and learn more both about the identity and also the quantity of different compounds in a sample.
Right, that's interesting. So you mentioned it's a way of sort of analyzing molecules. So how does MMR compare to traditional analytical techniques? And you mentioned NMR, for
Mhm.
example, IR, GC, or mass spec. How does it compare to those particular analytical techniques?
Mhm. Yeah, the way we think of it is it's characterizing different aspects of the molecule's
Right.
structure, right? In NMR, you're looking at particular nuclei and their spins. Infrared, you're looking at vibrational modes. So really the kind of the biggest, and I would say these are all sort of orthogonal techniques that really work together to give the most complete can give the most complete picture of
Sure.
a molecular's sample's characteristics. But really for us, we're really most sensitive to that three-dimensional structure. Subtle changes in, look at things like regioisomers, just the relative um, orientation of different atoms. While NMR, infrared, those are sensitive to those kinds of, those changes will certainly affect affect the what the spectrum will look like. In MRR, we're just very exquisitely sensitive. The connection between the total three-dimensional structure of the molecule and the data that we see on instruments that on BrightSpec's instruments is just it's connected in a way that exceeds that of other techniques to allow for really precise and highly highly selective. molecular identification.
Well, that's quite interesting. So what types of molecules or compounds are best suited then for the analysis with
Mm-hmm.
MRR?
So, being a gas phase technique, certainly where a lot of our initial applications and the history of the technique are rooted are in relatively small and volatile molecules.
Right.
And when we say that, we're talking about, and the complexity of the spectrum. It depends on things like the size, the amount of flexibility. So even for us, if the molecule adopts a different conformational isomer, so just a single bond rotation where the molecule can interact and it can exist in multiple states, we'll see that complexity in our spectrum. But it really works. It works well for molecules currently under around 300 to 350 AMU. That's sort of below that range is where the technique is currently really sensitive and really powerful. And molecules that we can get into the gas phase. We'll talk. I know we'll probably talk a bit more about kind of where the technique's going, but certainly at the moment, that small molecule volatile universe is where we're doing most of our work.
Right. Okay. And in terms of providing structural information, how does it work in terms of I always pronounce the word wrong, but is it chiral or chiral molecules,
Mm-hmm.
or distinction between stereoisomers? I mean, how does it work in terms of
Yeah.
that structural information?
Yeah, so I'll say in terms of things like constitutional, I'll set aside the enantiomers maybe to talk at the end of my response here, but a lot of other types of isomers, diastereomers, regioisomers, things like that. Um, the way we identify things, as I said earlier, that they have different arrangements of their atoms in space, so that will give them different spectral signatures. What we do is we're using modern quantum chemistry calculations to predict for a given structure, calculate that three-dimensional structure, and predict what our spectrum will look like. And we can do that now through developments over the last couple of decades with a really high degree of precision. So given a sample, without knowing for sure which of the isomers are present or if it's a mixture, we can record the spectrum on our instrument, analyze it, compare it back to those calculations, and say with a high degree of certainty which one we have. For enantiomers, for chiral analysis, what's been developed out of the University of Virginia and technology that BrightSpec is commercializing is the introduction. We can introduce a small, we call it a chiral tag, a small chiral molecule that will actually bind to the analyte of interest in the gas phase, sort of like a derivatization, but there's no wet lab work, and induce a structural difference between these compounds. And that's a really powerful, in an array of industries, being able to do that effectively is a really powerful technique that's really come in the last couple of years, and another dimension for the techniques for MRR's capabilities.
Well, that leads nicely. Actually, what I'm talking about is let's talk about applications within pharma
Mm-hmm.
specifically. So what are the most common use cases for MRR in pharmaceutical research and development? I mean, what areas can you use it for?
Mm-hmm. Yes. So some of the ways we've been using it, using our products over the last couple of years, and we've been fortunate to work with, collaborate with a number of companies, including publishing some papers to really demonstrate how this can be done, how this could be used. One of the starting points is in impurity analysis. So quantitative determination of potentially toxic impurities in pharmaceutical samples. That's obviously a common, an important need. And we've started honing in on the types of analytes, the types of impurities that might be difficult to do with other techniques, in particular GC or LC. That's kind of a starting point, a way for our technology to start to be adopted in the industry. We're also working on structural verification. So of course, in many industries, but particularly in pharmaceuticals, knowing the exact structure, knowing the precise which isomer is which, is extremely powerful. And we can provide a more confident orthogonal validation to NMR results, especially in cases where reference standards for all the compounds you're interested in might not be available. And then, continuing where we're really going with it, and we've published a couple of papers to show how this can be done, is to look at identifying and quantifying reaction impurities in a more confident and efficient way than can be done with other techniques. Again, with a focus on structures, impurities that might just be slight tweaks, of the molecular structure. But we know that when it comes to especially, medicines that we're going to take, those slight variants in structure can cause really dramatic differences in the effects those might have on our bodies. So of course the industry's really interested in knowing what impurities might be present, and then ultimately making sure that they're identified and safe before releasing the product.
Sure. Well, you mentioned GC, for example. So
Mm-hmm.
how does MRR complement or replace
Mm-hmm.
GC in industrial applications?
Yes. I think initially, like, I mentioned, I think initially what we want to do is help fill in some of the gaps where GC might be used or might not, but in either way, it might be a suboptimal, the suboptimal technique, and MRR can be a simpler, more quantitative, just easier to deploy option. Certainly down the, and in addition to the troublesome molecules, when you're looking at, GC most commonly coupled with either, a flame detector or mass spec, either way, MRR can help solidify the identification. What's the precise molecular structure that corresponds to each peak that might be in a GC chromatograph, and then to help, make sure that there are in a GC column, of course, two compounds might come off the column at the same time. They might coalute. That really complicates the identification, can lead to misidentifications. MRR can help solidify that and give greater confidence in the information that's coming out of GC and other techniques. So I think they work together really well. Certainly in terms of, we're doing a lot to demonstrate sort of cross compatibility, that the results from the quantitative performance of the two techniques is similar, and that we can really support by giving more concrete, more confident structural information.
Right. Okay. And well, let's talk about BrightSpec's technology in terms of supporting residual solvent analysis in
Mm-hmm.
drug manufacturing. So, how does that work?
Mm-hmm. Yes, that's a great that's a great example, or a great starting point, I guess, for us in the pharmaceutical industry, and one that we've deployed to a couple of pharmaceuticals now, and worked with different organizations on validating. Residual solvent analysis is just it's a ubiquitous, very common analytical need in pharmaceutical labs, to be able to validate that the pharmaceutical sample that's being released has a low enough amount of residual solvent. There's quite an array. There's something, approximately 50 to 60 residual solvents, and of course that are regulated by the different regulatory agencies in the U.S. and Europe and other places. Plus, of course, more solvents that are being used and still need to be evaluated. So over the last couple of years, we've developed a similar to the most common technique that's validated now as a GC headspace technique.
Right.
So we have a similar, MRR headspace technique, which is a very familiar sample preparation procedure. It's essentially, you have your drug product, and you dissolve it into a in some sort of solvent diluent to allow a headspace to form. And then instead of injecting through the GC, we put it in you put it in the MRR autosampler, and the instrument performs quantitative analysis on a panel of solvents of interest to you. I think what's most exciting for us is we've validated that the sensitivity of the instrument can meet, we've demonstrated it can meet the requirements of the regulatory bodies. It works with the exception of just a couple of solvents that don't have the required functionality for us to detect them. It's a pretty universal method to both high volatility as well as low volatility residual solvents. That's certainly, when you look at, comparing to GC, that's one area where, our technique can be a little bit more universal. And then the other element to it, which maybe upfront talking about the comparison to GC, we're not doing chromatographic separation. We're not
Right.
we're not taking our system through a column. The volatilized sample flows right into the instrument. So the speed of the analysis, even when the sample is pretty complex, that the speed of that analysis can be, seconds to minutes rather than, the time 15 minutes or longer that can come from a GC separation. Really, all that in MRR, the only thing that affects how long your measurement is going to take is the components you want to measure and the sensitivity you want to go to. If you want to go to low level sensitivity, you can spend more time averaging. If you want, quick determinations, those measurements can be really fast because it's just a direct analysis.
Right. And let's talk about astrochemistry then, because I know it is
Mhm.
also used in astrochemistry research as well. So can you explain more about that?
Absolutely. Yeah. So this kind of where the history of the history of the technique, before BrightSpec. So this technique dates back to the 1940s. It's been around. The fundamentals of it. The research groups around the world have been working on this technique for a long
Right.
time. And one of the areas, from the beginning, where it was of the most there's the most activity, is in studying molecules in distant star forming regions. And the reason that really I'll say rotational spectroscopy and characterizing molecules through the rotational modes. The main reason that's the technique of choice is because the you know stars are born in dense clouds of dust and gas. So molecules, including complex organic molecules, are forming in these regions that are just very shrouded. So looking through from an optical telescope or even in the infrared, all of the dust just completely obscures the view. But when you go down to this radio frequency regime or millimeter wave regime, the frequency range where BrightSpec's instruments are operating, light can penetrate through those areas.
Right.
Also, as we've been talking about, each molecule present, including especially when there's really a lot of structural diversity, each molecule has highly characteristic signatures that encode a lot of information about what the molecule is, its abundance, all kinds of interesting information. That radiation can reach the United States, or can reach the Earth, and reach radio telescopes and be analyzed in much the same way as we're doing that. The reason I bring up the United States is to say also about Charlottesville. This is really a hub. In addition to the University of Virginia and the research in MRR, this is really a hub for radio astronomy here. The National Radio Astronomy Observatory is headquartered in town, and here they manage resources. The telescopes located across the world, where researchers still can apply for time to measure the rotational spectra of distant regions. And before I joined BrightSpec, some of my work was in using those telescopes and also helping to develop laboratory instrumentation to help support the analysis of all the data that they're starting to measure. It's a really
R-
exciting area for
I can
the technique.
imagine it. Yeah, sure. Now you mentioned obviously you've got some white papers and you also work with some customers. So, um, what case studies have you got to sort of illustrate the value of MRR,
Mhm.
particularly within the pharma space?
Yes, absolutely. So we've published a lot of the I would say a lot of the central themes have been around um, showing that we can quantitatively analyze mixtures of isomeric compounds. That's been a lot of the theme that has um, and several of the papers, and some of those have been in more of a uh, for starting materials, raw materials. We have a paper. One is more in a online reaction context. a couple now that have shown how we can do these measurements in a more accelerated fashion and an automated fashion. A couple with chiral analysis. Those have been some of the themes. We've also been in the last couple of years working on demonstrating sort of one of the developing applications of the technique, which is for isotopically labeled substances. This is, deuterated drugs are coming on the market, and being able to precisely determine the position at which isotopic modifications are being done is certainly a unique capability of the technique. Yeah, I think we've been fortunate in terms of the collaborators we've gotten to work with who have, I think there's a lot of coherence between the different applications, but there's also some differences in terms of the types of compounds we're studying, of course, but also just what the exact needs of the analysis are. And it's been really good to be able to publish and put out in a peer-reviewed journals for interested researchers and interested customers to read and understand more about how the technique works and how it can be used.
Fantastic. Well, look, we'll put some links to those white papers hopefully above this video as well. Now, before we talk about the future of MMR, I was thinking as you were talking about the comparison with GC, and obviously they use chromatography as well as part of the process to split it. If someone's listening to this and they've never seen an MRR or one of your instruments, how big, and it's hard to say on a video when you haven't got anything in front of you, but
Yeah.
how much bench space does it take, for example, to a typical GC setup to do some of the workflows you were talking about a bit earlier?
Okay. Yeah. So our sort of our main product, or one of the main products we're going to be launching here coming up in the next few weeks. It's on a cart. It's about a 4 feet in length, and then 2.5 feet depth. It's meant to be, it's portable. It can be transported around labs. It's on wheels. Has a simple gas power requirements. It's meant to be. It's similar. That instrument doesn't sit on a bench top, so it takes up a little more space maybe than a traditional GC. But in terms of sort of the lab space it occupies, it's about the same form factor. I will mention we are also launching a smaller benchtop product as well for educational and a little more targeted industrial applications, called the Nanomer, which is a benchtop very similar. It's about the same bench space, maybe about 4 feet in length, but can sit on a table and again takes up about the same space as a typical GC mass spec system.
Well, that's interesting. Well, that's a lovely segue. My next question was going to be, what innovations have you got in the pipeline for BrightSpec's MRI technology? I think you've covered that to a large degree just in your answer there. Now, looking ahead though, how do you see the technology evolving, over the next few years, in terms of both the academic setting and also you mentioned the industrial setting as well?
Yes. So, I mean, I think really, the way we look at things, there's sort of two main areas we want to continue to push the technique beyond, in the next couple of years. One of those is, of course, the range of applications that we can address, and a lot of that comes around being able to handle, for the instrument to be able to handle more complex mixtures. Mixtures at lower level, lower concentrations, lower sample quantities, all those things that really matter when you push, especially in the pharmaceutical space, but also in other areas too. Constraints that analytical chemists are used to dealing with. We want to keep improving our specifications there. Sampling increasingly complex, low volatility molecules. So a lot of BrightSpec's innovation and sort of our intellectual contribution to the technique, in addition to the manufacturing design, has been around the sampling technology, making it easier and more accessible to introduce samples into the instrument. And then the other area of it, of course, MRR is a technique that outside of the research community is still fairly new. and a lot of the interpretation of the spectrum can be, certainly have learned over the years, can be one of the most intimidating aspects of the process. And that's a lot of the publications we've done have been cases where, the users have provided this, the collaborators have provided the samples, and really BrightSpec's, expert scientists have done the spectral measurement, done the analysis. Now where we're really transitioning is to make it so that users, fairly with a fairly low degree of training, basics of how to operate the
Sure.
instrument, operate the software, but without having to, spend a couple of years studying the quantum physics all the intricacies, really automate and simplify that analysis process. And I think we've made a lot of strides there, that we're really excited about, but we want to keep pushing the envelope on, again, complex spectra, mixtures, chiral analysis. These are areas where, um, we think over time will really lower the barrier to entry, and help, hopefully, make this technique something that really, just like GC and MR are today, that anybody with a basic background in instrumentation can approach, and really get to use the power of the technique.
I think that's a very good point, because if you, for example, go to ASMS
Hmm.
every year, what you'll see is that they've simplified
Hmm.
the instruments to make it easier and easier for traditionally non-scientists to use as well as
Hmm.
scientists, so you don't have to have that background. That's a very good point. Well,
Hmm.
that's fantastic, Justin. Thank you for that overview. I've learned a lot more about MRR. I hope everyone else who's watching has as well. Now, obviously, you mentioned some things that you're launching. Actually, you're going to be at PitCon, aren't you, at the beginning of March in
Yes, yes,
Boston,
we sure are.
where you are actually announcing a lot of these launches, I understand. So, viewers, if you are attending PitCon in Boston, then make sure to check out BrightSpec's booth. I'll put the details again above this video with the stand number so you can find that. And we'll put the links to, obviously, BrightSpec's website and the white papers that Justin mentioned. But also, if you've got any questions related to what we've been talking about today, and particularly in terms of comparisons to some of the other technologies that Justin's comparing MRR to, then feel free to put them in the comments below, because I'm sure he'll be only too happy to answer any questions you've got as well. So, Justin, it's lovely to meet you. Uh, enjoy Charlottesville, and hopefully I'll maybe see you in Boston for PitCon as well. But have a fantastic 2025 year. It's been lovely to meet you. Thank you for sharing all that information with me. And viewers, as I said, I hope you found that interesting. If you've got any questions, please feel free to put them in the comments below. And you can meet BrightSpec at PitCon, as I said, and in a few weeks' time. It's frightening thing is we're already getting towards, I mean, we're in the middle of January. Beginning of March is not that far away, really, when you start thinking about it. So you can see them then. So, Justin, thank you very much for your time. Viewers, thank you for watching. Have a fantastic 2025. Until next time, as always, stay safe. Bye bye.